When you think of air filters, spider silk is probably not the first thing that comes to mind. Recent research published in Nature Communications could change that. Scientists developed spider silk-inspired fibers that can capture bacterial bioaerosols, which may pose a risk for human health, with nearly 100% efficiency. The results could be useful for advancing the development of air purification and filtration systems. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. So to begin, let's talk a bit about bioaerosols, what they are and why they matter. Bioaerosols are tiny airborne particles that are derived from living organisms, including plants and animals. They can include pollen, dander, and droplets that contain microbes like bacteria, viruses, and fungi. Because of their small size, bioaerosols can travel between environments relatively easily and may remain suspended in the air for extended periods of time, depending on conditions within the environment, for example, how stagnant the air is. Bioaerosols have various health implications. Inhalation of bioaerosols can lead to infection and disease depending on the bioaerosol and the host. This is something we've all become highly aware of during the COVID-19 pandemic, given SARS-CoV-2, the virus that causes COVID, spreads via respiratory droplets released from infected individuals. With that in mind, air filtration systems have been and will continue to be important tools for managing bioaerosol exposure and potential risks. To that end, scientists are developing innovative methods for capturing bioaerosols, and they're looking to spiders, of all things, for inspiration. Which brings us to a new study in which researchers developed fibers based on the structure of spider silk to capture and inactivate bacteria aerosols in the air. Of course, one wonders, why spider silk? Spider silk is a strong, sticky protein fiber. Structurally, it consists of fibrous spindle knots separated by joints, sort of mimicking beads on a string. This structure makes spider silk good at capturing particles and moisture from the air. Essentially, water latches onto the joints and the silk and begins to pool around the spindle knots. Researchers have previously capitalized on the water capturing capabilities of spider silk, developing artificial fibers that could be used to collect water from the air, potentially in places where water may be hard to come by. However, the utility of these fibers for capturing bioaerosols containing microbes has never been explored. So that's what the researchers in this study aimed to do. To start, they created nylon fibers dipped in titanium dioxide solution. The titanium mixture forms small droplets that shrink and dry, creating joints and knots that mirror those in natural spider silk. Now, the choice of titanium dioxide here is an important one, given this compound is a photocatalyst. Photocatalysts are activated by UV radiation to produce reactive oxygen species, or ROS. These ROS can interact with other compounds in water and air to produce water and other byproducts. They are useful for various purposes, including decomposing pollutants. ROS generated via photocatalysis can also interact with cells, including bacteria, and lead to cell death. With that in mind, the scientists wanted to couple the antimicrobial powers of photocatalysis, that is, the titanium dioxide, with the water-grasping structure of spider silk, a combination that had never been attempted before, and the scientists hypothesized could be useful for controlling bacterial aerosols. So did it work? The researchers examined the ability of the artificial spider silk to capture bacteria using E. coli and Bacillus subtilis as model organisms. They positioned the silk fibers in front of a UV light to activate the photocatalyst and then sprayed the bacterial suspension on the silks. As observed here, just like in natural spider silk, the droplets formed on the artificial spider silk. The liquid accumulated in the joints and then moved into the spindle knots, freeing up the joints to collect more of the bioaerosol. And wherever the droplets went, the bacteria concentrated there as well. The bioaerosol capture ability of the silk was tied to various factors, including how fast the bioaerosol was coming at the silk, the concentration of the bioaerosol, how big the spindle knots and joints were, and more. The scientists also wanted to determine whether the bacteria were not only captured, but also killed. They showed that in the absence of UV light, bacteria grew on the silk. However, in the presence of UV light, bacteria were completely inactivated after 4 hours. That is, after this time, the bacterial load on the silk decreased from 10,000 colony-forming units, or CFU, per 5 centimeters to 0 CFU. Even when the bacterial stock contained 1 billion CFU per milliliter, the spider silk photocatalyst achieved a 99.99% inactivation efficiency. The morphology of the bacteria was noticeably altered after photocatalysis as well, as its cells became small, cracked, and flat. Through additional experimentation, the researchers proposed a two-pronged mechanism for how the artificial spider silk kills the bacteria. Essentially, reactive oxygen species present in the water droplets attack the bacteria while the silk is still moist. Then, once the droplets dry, the ROS at the air photocatalyst interface further inactivates remaining bacteria. So, will we be using spider silk-based air filtration systems in the near future? Not quite, as this study is simply a conceptual stepping stone. The fibers discussed here in and of themselves are not a full-blown air filtration system. However, the results are intriguing in light of currently available systems. Commercial air purifiers capture bioaerosols in several ways, as shown here. Exhaust systems associated with air filters are effective, though can be blocked and consume lots of energy. Moreover, such systems capture but do not inactivate bioaerosols. This means that microbes can remain viable or proliferate within the system, potentially making the filter itself a source of contamination. Here, the scientists created a new method that does not rely on an exhaust system, thus is low energy and overcomes other limitations associated with those systems, and can kill bacterial bioaerosols, thus reducing the chances of contamination. So let's summarize what we've covered today. For one, bioaerosols can pose a risk to human health. Air filters and purification systems are useful for limiting bioaerosol exposure in certain settings. However, current systems have some limitations, including high energy consumption and an inability to kill microbes in the air. To overcome these limitations, researchers developed spider silk-inspired fibers that effectively captured bacterial aerosols and achieved an inactivation efficiency of nearly 100%. The findings provide a useful foundation for developing new, efficient, and effective air filters for managing airborne microbes. And that's all for today. Be sure to subscribe to ASM's YouTube channel and enable notifications to be notified whenever a new Microbial Minutes drops. I'm Madeline Barron. I want to thank you for listening, thank Ray Ortega for production, and I'll talk to you soon.